Good morning, ladies and gentlemen. I'm James from the Chinese University of Hong Kong, and I'll be your student host today for the webinar. Before we begin, I would like to express my great thanks to the Hong Kong Life Sciences Society for organizing such a fruitful webinar. We are very pleased to have Professor Tsui with us today. Professor Stephen Tsui is a professor at the School of Biomedical Sciences in the Chinese University of Hong Kong. He is very interested in bioinformatics, comparative genomics, and molecular biology of clinical pathogens. Indeed, during the SARS outbreak, his team was one of the earliest teams that cracked the complete genome of SARS coronavirus. And today, he'll share insights into the characteristics of the SARS coronavirus genomics and also the molecular epidemiology of COVID-19. After that, we will have an open Q&A session. Please be reminded to follow the instructions on participation guidelines regarding how to ask a question. Remember to click raise hands on the participant panel and keep the icon on. I will then introduce your name when it's your turn to ask questions. And please be reminded, one question at a time. So now let's welcome Professor Tsui.
Hello, good morning, everyone. I will try to share the screen. So I'm wondering whether you see the PowerPoint slide.
Yes.
Yes, oh, that's fine. Okay, so I will try to start my presentation. Thank you very much for the invitation from the society. So today I will share something about the SARS-CoV-2 genome and the molecular epidemiology of COVID-19. So basically, I would tailor the talk to be more educational, just because I know there are lots of students here. So that I hope you, after the talk, you know how to do the research and also how to understand the outbreak. In fact, 17 years ago, I cracked the SARS genome. So this is my team at that time. Eventually, we visited by Mr. Tong, okay, the chief executive at that time. And then these are also a memorial photo. Professor Joseph Song actually is the doctor who treat the patients. I work on a viral genome sequencing, and this is a patient, okay, of SARS, also a doctor in the Prince of Wales Hospital. So this photo had been taken 10 years after the SARS outbreak. So I think you know Professor Yuan Guoyong as well as Professor Joseph Song. Okay, but in fact, a lot of people here are involving in the COVID-19, for example, in Wuhan, Shek Jinglai, and also in Beijing, Gou Fuk. Okay, so many of them are now working on the current outbreak. Okay, so basically similar team. Okay, go back to the structure. So before you know SARS-CoV-2, okay, first of all, you need to know something about SARS-CoV, okay, because they are very similar. The SARS coronavirus actually is a very simple virus composed of the RNA, the nucleocapsid protein, the spike protein, and the membrane protein. So basically, the virus will bind to the receptor. Okay, for SARS-CoV, it is ACE2. Also ACE2 for the SARS-CoV-2. And then the RNA will enter the cell, produce a lot of proteins and RNA. And then after repackaging, it will leave the virus and lead to the cell rupture. And then those viruses will infect other cells. So in the last outbreak, in Hong Kong, there are not too many patients. Total death case is only 298. So why we need to decode the SARS coronavirus? Okay, why the decoding is so important? There are several reasons. Okay, first of all, we need to have a very quick and accurate diagnostic test. And then we need to trace the mutation of the virus, we trace the origins, and prepare vaccine and antibody for prevention and treatment of SARS. So I still remember in 2003, we only have the genome of the coronavirus 229E. Okay, so at the very beginning, people are still using the genome of the 229E for the detection of SARS-CoV. But obviously, it is inefficient. Okay, so using a wrong viral genome to detect a virus. Okay, basically, just like you take a photo. Okay, you pick any photo and then you try to catch a suspect. Okay, so you will fail certainly. Okay, you need to have the right genome in order to detect the right virus. So we start our project in 2003, okay, in a team, okay, so all the students are well protected at that time when they do the experiment. So for the coronavirus, the genome is pretty big. It is around 30,000 nucleotides. It is a positive strand RNA. So how can we get the genetic code for this 30,000 nucleotide? So at that time, we get the virus, we extract the RNA, and then we try to amplify the DNA using polymerase chain reaction, and then do the sequencing. This is the result of the PCR bands. And then at that time, we use a sequencer called a megabase, okay, that can sequence 96 samples within 2 hours. And then we have a lot of fragments from the sequencing. Okay, the banding is like this, okay, different colors represent different nucleotide. So after we get a lot of fragments, we can use bioinformatics to align the fragments into the full genome. So that's what we have done. So finally, we published the paper in New England Journal of Medicine. In fact, at that time, we used 13 days, okay, to finish the first genome. Okay, totally, I involved 14 students and staff in the sequencing. When we look at the SARS-CoV genome, it composed of many open reading frame. Okay, so the most important open reading frame is called the S protein. That means the spike protein. So this protein binds to the receptor and then trigger the entry into the lung cells. But there are also a lot of accessory proteins, just like envelope protein, the membrane protein. So those are also important proteins. So how can we trace the ancestor of SARS-CoV at that time? I would like to show you this figure. For example, at that time, we sequenced a lot of genomes in different places, three in Beijing, some in Guangzhou, some in CU, that means in Hong Kong, Canada, and US. Okay, so this is the position in the viral genome, and this is the nucleotide. For example, for the first one, okay, at this position, okay, different nucleotide can be observed in different strain. But if I ask you what's the ancestral nucleotide for this position, certainly you will tell me it should be G, okay, because it's common, okay, quite common in most strain. But in this strain, okay, finally it mutated to T, okay. So for the second, it should be C. Eventually, you will get this, okay, the sequence, what we call the ancestral sequence. And then when we compare this ancestral sequence with all the sequence, then eventually we will know Guangzhou is the sequence that are most similar with the ancestral sequence. That's the reason why from this, we can assume that the outbreaks originated in some place near Guangzhou. Okay, so this is the method we used to trace the origin. So based on this method, look at the mutation when the virus spread. Eventually, we map the whole story, how the virus spread to Zhongshan, Guangzhou, Shenzhen, and then to a hotel, okay? We build a whole story based on the virus mutations. I've also worked in the Amoy Garden outbreak. At that time, this very terrible. I think recently, in the last few days, there's also another outbreak in Guntong, okay? So very similar, okay? So we look at the patients in different floor and get the samples and then do the sequencing for these patients. And then we can study who is the index patient, that means the first patient, and how they spread to different floors, okay, based on the sequence, okay? That is what we call the molecular epidemiology, okay? And then we published the result in Lancet at the time. So eventually, okay, just 1 year after the last SARS outbreak, we published a paper in Science telling people how to use this method, look at the mutation, and build the whole story of the viral spread. Okay, so eventually this becomes the standard method people using, okay, for outbreak tracing. So basically what you see recently is originated in Hong Kong. And also at that time, we developed the real-time test for SARS-CoV. Okay, so normally, before the SARS outbreak, normally people use virus culture for the detection of virus, okay? But starting from the SARS outbreak, okay, because of the genome sequencing, we can design a pair of primers and specifically amplify the virus, then that becomes the standard method afterward, okay? In the last 16 years, in every outbreak, people use real-time PCR for the detection of the virus, okay? So this is also originated in Hong Kong. So you see how important is the role of Hong Kong in infectious disease control. So this is an old slide that I present in 2007. That means 13 years ago. So at that time, I asked my student, will SARS come back? Okay. So my answer is SARS will come back one day. There are a very important reason. Just because the host of the SARS coronavirus is bat, you will ask me, why bat? Bat is very special, okay? Bat is mammal, okay? A mammal, okay? That means closely related to human. And also bats can fly, okay? The only mammal that can fly, okay? So that means those viruses that can infect bats, because bat is mammal, can also infect human, okay? Because we are closely related. We are also mammals, okay? And also bats can fly. That means they can hide anywhere in caves, okay? For example, you can never get rid of bats, okay? So those viruses in bats, those coronavirus in bats can jump to other animals, okay? And then cause another outbreak, okay? So I can forecast this will not be the last one. There will be future outbreak of SARS, okay? So MERS is another coronavirus outbreak that happened in 2012. So this is from Middle East. So let's see COVID-19, okay? So we move to the topics today. So, but from that background, then you know quite well, okay, about how to look at the information in the following slide. 17 years ago, we still use capillary sequencing, what we call the Sanger sequencing, okay, to sequence the virus. It takes a lot of efforts, okay, a lot of manpower, and also a much longer time. But in recent years, because of the development of the next generation sequencing, now we have very powerful sequencer, or we can even use a third generation sequencing. Those are handheld machines, okay, that can sequence the virus, okay. So basically, once we have the viral culture, we can get the genome within 1 to 2 days, okay, very quick. Basically, nowadays in a lot of countries, okay, they can already sequence the genomes easily, okay. So that's the reason why there are also already thousands of genomes on web, okay, with for the SARS-CoV-2 just within several months. When we look at the genome of different coronavirus, okay, this is the SARS-CoV, okay, the virus 10 years ago. This is the MERS coronavirus, and this is the SARS-CoV-2. Okay, so when we look at the genome, basically the ORF1, very similar. Okay, all have the ORF1. So those are very important housekeeping proteins. So basically, they are very conserved. Okay, and then the other open reading frame, very important open reading frame, is the S protein. That means the spike protein. So you can find a spike protein here for different genomes. Okay, these two are the bad virus. They are closely related to the SARS-CoV-2. So when you look at this, what we call the accessory proteins. Okay, so those proteins could be quite different, although you can still find E, M, and N in nearly all genomes. But for example, I use the open reading frame 3 as an example, okay, because I'm the founder of the 3A in SARS-CoV, and I characterized this at that time. So when you look at 3, okay, basically, in MERS, there's 3. In SARS-CoV, it is 3A and 3B. But in SARS-CoV-2, it is 3. Okay, so when you look at other proteins, some may only present in some virus. For example, the 13 and 14, okay, these two are newly emerged open reading frame that cannot be found in SARS-CoV and MERS genome. Okay, so in fact, these proteins could be related to the pathogenicity and the severities, okay, of the virus. For example, I have discovered that the 3A is related to the apoptotic effect of the virus, okay, that causes a cell death in the lung, okay. So whether 3 have a stronger or weaker effect, okay, on lung cells, people are still working on it, okay. So this is the comparison of the genomes. But based on this, we will know more about how to treat the patients, how to study the pathogenesis of the viral infection. Okay, so this is very important basic information for further analysis. So we can also look at the distribution of patients in different provinces in mainland China. Okay, so of course the higher number will be in Hubei. But when you look at other provinces, you will know that basically those provinces that are more populated, okay, that means with a higher population density, they will have more patients, okay, just because there will be more social contacts, okay, that can spread the virus. You may also find this in a lot of papers, okay, so I can interpret, I try to help you to interpret this. So how to build this? We can easily build these phylogenetic trees using the genome. So when we look at this, we will know, okay, this SARS-CoV, that means this caused the outbreak 17 years ago. This is the SARS-CoV-2. Basically, they belong to the same branch and in the same lineage, okay? But for MERS, actually, it belongs to another branch, okay? It is quite different from the SARS-CoV, okay? That's the reason why MERS-CoV are much more pathogenic, okay? The death rate for people who contract the SARS-CoV is nearly 50 to 70%, very terrible. The pathogenicity of this is very high, okay? Another branch which composed of the 229E, okay? So this branch is OC43, okay, this branch is much less pathogenic, okay? So based on the location, okay, location of the virus in the tree, basically we will know, okay, whether it is more pathogenic or less pathogenic. Okay, so basically because SARS-CoV and SARS-CoV-2 are in the same lineage, they don't have a very big difference in the pathogenicity. Or we can say the symptoms for patients with COVID-19 is a little bit less severe than that with the SARS-CoV. Okay, so so you may also find figures like these in a lot of websites. So how to build this? Basically, we can use the phylogenetic tree here, and then input the information, okay, of the virus. Okay, two sort of information. Okay, the location of the virus and also the time. Okay, for discovery. Okay, so basically we can put this, okay, information with the tree, then we will know how the virus spread. Okay, so when we combine these, then we can rebuild the whole story. Okay, and rebuild what we call the dissemination dynamics of the virus. And also when we look at the genome, we may also explain, okay, why SARS-CoV-2 can infect some species, but not the others. Okay, so here is the comparison of the ACE2 of different species. Okay, as I have mentioned, ACE2 is the receptor for SARS coronavirus as well as SARS-CoV-2. Okay, so human, of course, okay, will be the host. So pig could be infected because the main difference is only it's not too many. Okay, they're closely related. Cat, okay, can be, okay, also chimp can be, okay, can be the host. But for other species, if they are less similar, then the chance that the virus can infect the species will be lower. Okay, so whether a species could be infected basically depends on the variation in the ACE2. Okay, so and also people now also use the structural analysis, okay, to explain why SARS and SARS-CoV-2 can infect some species but not the others. Regarding the treatment, basically the most useful treatment will be surveillance. Okay, how to isolate the patients, okay, how to restrict the social contacts. It is the most well-known method, okay, and most powerful method, okay, because the viral spread is so quick. Someone asked me, okay, whether vaccines is useful, but I can tell you, okay, basically from research to distribution, it takes around 200 to 300 days, okay, to have the vaccine. So people now forecast that the vaccine will appear by the end of this year or early next year, okay? So whether vaccine is useful depends on whether the outbreak still exists, okay, by the end of this year or early next year, okay? Just like SARS, it disappeared, okay, just within a few months, okay? We can never predict whether the virus will disappear, okay? Of course, now the most recent understanding will be this outbreak, okay, will continue next year, okay? Just because it already moved to those countries in the southern hemisphere, okay? Just like Australia, just like Brazil, okay? Eventually, they will spread the virus again back to the north hemisphere. So, if you ask me, what is the beneficial? I try to avoid this, but I can say this, okay? Beneficial effects of this outbreak. I can tell you, starting from the pandemic in 1918, okay, very terrible, the pandemic. Basically, around 200 people report that around 200 million people died, okay? So this is more conservative, okay, 50 to 100, because that is only the record. But at that time, we don't get the right number, okay? The number should be a little bit bigger than this, okay? This is the first influenza outbreak we record, okay? So, but when you look at this, okay, we can say every 30 to 40 years, there will be a very serious influenza outbreak, very serious, okay? So, for example, in 1968, this is called the Hong Kong flu, okay? The last big outbreak, okay, before the COVID-19, okay? So why the separation is always around 30 to 40 years in the past? Just because for the first outbreak, a lot of people survive. Why they survive? Because eventually they have the antibody against the virus, okay, the virus circulating at that time. And eventually, okay, this antibody, okay, will protect them, okay, in the next 30 to 40 years. Until another generation emerge that don't have the antibody, okay, then they are very vulnerable to the outbreak. Then another outbreak will come, okay? So this prediction, 30 to 40 years, is very correct, okay? When you look at this, then the next outbreak should be 1998, am I right? 1998. In fact, it come back, okay? When you look at the composition of the influenza virus, they are all involved avian influenza. So why avian influenza is so pathogenic? Because avian influenza virus is quite different from human virus. Then it will become, just because of the big difference between human influenza and avian influenza, this will cause a big immune response, very severe immune response, and cause the death of the patients, okay? So every outbreak involves avian. So if you still remember, the last avian flu, okay, the big avian flu, okay, could be found in 1997 in Hong Kong, okay? So, but fortunately at that time, we tracked down the outbreak by our public control, okay? So eventually that have not spread to the whole world because of the efforts in Hong Kong. But I need to remind you, okay, it will come one day, certainly, it will come, okay? And also, just because avian flu is very pathogenic, and now the spreading rate, okay, when compared with previous outbreak, the viral spread could be 10 times or even 100 times faster than 50 years ago, okay? So the next outbreak will be extremely terrible, okay? So then you may consider this COVID-19 a rehearsal of the future avian flu, okay? So that we learn the lesson, and then in the future avian flu outbreak, we will have better control. Just like recently in Hong Kong, okay, why we have better control? Just because we have the experience 17 years ago, okay, we can learn, okay, from the outbreak, okay. I hope the whole world can also learn from the outbreak, although it is very painful, okay, but it will help us to fight against future viruses, okay. Actually, this is an endless war against the viruses, okay. Thank you very much. Your questions are most welcome.
Thank you so much, Professor Tsui, for the excellent sharing on COVID-19. Now we will move on to the Q&A session. Once again, remember to click raise hands on the participant panel and keep the icon on. I will then introduce your name when it's your turn to ask questions. So maybe I'll start first. As known, COVID-19 is more contagious and less lethal compared to SARS, while a recent scientific study has suggested that the COVID-19 virus has mutated into a more aggressive form. Can these phenomena be explained by looking into the genome and the molecular epidemiology of both viruses? Thank you so much.
So maybe I can tell you a dogma of infectious disease, okay? The more pathogenic okay, of the virus, the less will be the prevalence of the virus. That means if the severity is very high, okay, then we can easily identify the virus, and the virus may also kill the host. Then the chance it can spread the virus will be lower, okay? Just like MERS, it is very pathogenic, and eventually all the patients die, okay? And then it cannot spread easily. But for SARS-CoV-2, it is less pathogenic than SARS-CoV, okay? So a lot of patients could be asymptomatic so that they can spread the virus before they have the symptoms. That's the reason why the spreading is much wider for COVID-19 when compared with SARS 17 years ago. Another good example would be I can use Ebola virus. Extremely pathogenic, am I right? But just because it is extremely pathogenic, we can contain them a little bit easier because every patient we can identify easily, okay, based on the phenotypes. Okay, so then if you ask me when we compare this, I can still insist that COVID-19, okay, is the symptoms are less severe, but finally the consequence is much bigger. Okay.
Okay, thank you so much for
You're
answer.
welcome.
Yeah, so just a reminder that you have to click the raise hand on the participant panel and keep the icon on. And so I can actually introduce your name afterwards. So I'll also do the second question where I've actually read through the news that Professor Tsui, you are actually collaborating with Prenetics to launch the Project Screen, which is a non-profit initiative to provide testing and diagnosis for COVID-19. So I do really wonder what are the benefits that you would expect to gain from the project to the collaboration, and how would these benefits influence the society academically and socially? Thank you so much.
In fact, this effort is also suggested and assisted by the government. Okay, so what the government want to do will be, there should be a public-private collaboration. Just because we are playing different role for the public sector, for example, the CHP, Center for Health Protection, they do a lot of testings for suspects, okay, in the communities, in outbreak, okay. So those will serve the public health control and the healthcare system, okay, more clinical. But in fact, there might be private sector that serve someone that may not have the symptoms already, but they want to know whether they have, okay? And also in the near future, okay, if we open the border, okay, then there will be a lot of people move, okay, around. Then the government lab may not have the capability to do all the testing, okay, for those travelers, okay, because the government is not that scalable, okay. They are hosted in the clinical setting, okay. So that's the reason why the government also encourage private labs, okay, to launch their service, okay. So Project Screen is one of the project, okay, initiated by Prenetics. I'm the chief advisor of Prenetics. That's the reason why I assist them to build the test and do the accreditation.
Okay, thank you so much for your answer. So we have a question over here from Fule Wuchu, and I will unmute you so you can ask your question, sir. Oh.
Can you hear me?
Oh, yes.
Yeah. Hi, Professor. So I have a question about how to find the ancestor sequence of the virus. So during the presentation, you tell us that so we find that at each position. So the most common nucleotide is the nucleotide that appears in the ancestor virus. But why this is the case? What if the virus has already mutated a lot in the population? So why is the most common nucleotide is the ancestor sequence?
Oh, okay. So we assume that, okay, for the first virus, there should be a sequence, okay? And when it spread, the viral genome will replicate, and because of the replication errors, mutation will emerge. Okay, so that means for virus, there are more mutations. They will be the later virus. They will not be the early virus. Okay, and then based on this, what we call the convergence. Okay, convergence of the sequence. Okay, finding a common sequence. So the most common sequence will be what we call the ancestral sequence. Okay, this is ancestral sequence. So basically, we find this. Okay, very similar. But Beijing sequence, okay, they're quite different. For example, this one quite different, okay? But this is most similar, okay? So that's the reason why we say this is, of course, I have simplified the picture. Normally, we use software to calculate. We use very complicated statistics software to build the model like this, okay? But the rules is like what I've mentioned, okay? Based on the emergence of, so just like if I can say for this branch, if for this branch, okay, for the first virus, a particular position mutated from G to T, then all of them will have the mutation of G to T, okay? That will become the fingerprint of this cluster, okay? So just like detectives, we can base on mutation, and then we know what happened to the viral spread. I hope this is not that complicated, okay? That's the reason why I use this figure to illustrate the situation.
Okay. So thank you so much for asking a question. I was answering a question. So there is another people who asked a question, GYG. I'll unmute you so you can ask your question.
Thank you very much indeed. Prof, thank you for a lovely talk. It was a pleasure to hear someone who's been working at the front line. I'm a cell biologist. I'd like to ask you, please, with the variation in the genomes that you spoke about and the differences in lethality, how applicable are cell biology studies on SARS and MERS and extrapolating from those to COVID-19? And then the second part of the question is, what would you say would be the value of a model system, be it in vivo or in vitro, to COVID-19 studies?
Okay. I answer the first question first. The first question, I saw maybe this slide. So once we have the genome, then as I mentioned, we have more information. A lot of research have been done on SARS-CoV and MERS-CoV, okay, all these proteins. So some of the knowledge could be transferred to SARS-CoV-2, but we need to look at whether they have the same open reading frame or whether they have the same sequence, okay? Some could be transferred, okay? For example, 3, okay? I'm still looking at this moment, okay? But of course, new experiments should be performed to validate, okay, on the SARS genome, okay, on cells. So I also work on molecular biology as well as cell biology. Okay, so we are now cloning the gene, okay, to characterize the gene. Okay, so this will be my answer. Okay, some of the knowledge could be used. So I forget the second question. The second question is.
Sorry, Prof. I was going to say, but I think you've answered what the value of the model systems, be they in vitro or in vivo. Yeah.
For in vitro, the most traditional cells people use, if it is non-human, will be the monkey viral E6 cells. But now a lot of people also use a lot of human cells, just like human lung cells, epithelial cells. For model, the most recent, of course, we can use chimpanzee, but I think it's not feasible. People are suggesting that hamster will be the most convenient in vivo model for the virus, because SARS-CoV-2 can infect hamster, and the symptoms are quite similar to human.
Okay, thank
That's
you.
great. Thank you very much, Prof. Thank you
You're
indeed.
welcome.
Okay, thank you so much for asking a question and answering a question as well. So I'll also ask a question that is a common question among people that how can the findings of COVID-19 epidemiology be placed in a larger context to assist in the treatments, probably diagnosis, and also assure the well-being of the patients? Thank you so much.
Okay, so in fact, we can assume that the virus will continue to mutate. Just like when we look at the situation, okay, the clinical situation in different countries, they may have different symptoms. For example, in UK, okay, they mentioned that the children may have very different presentation, okay. So just like during the last SARS outbreak, in the early outbreak, patients only have lung problem. But eventually, in later outbreak, they develop GI problem. Okay, this could be caused either by the routes of transmission, okay, how they get the virus, or caused by the mutation of the virus. Okay, that may also trigger different phenotypes. So we need to closely monitor the viral genome, okay, and look at, okay, the relationship between the genome and the phenotype, okay. So based on this, we'll have the idea which proteins lead to a particular phenotypes, and then this may give us some idea how to treat the disease, okay. So although a bit indirect, just because for the genome, okay, normally we only use it for control more than treatment.
Okay, thank you so much for your question and also answer. So I saw a question from the chat panel that since it actually mentioned that Professor Tsui, you had a lot of experiences in SARS and also in coronavirus, and it's a wonderful speech about how it's actually being linked to MERS and also other diseases. And he wants to ask what are the major challenges and difficulties as a researcher when you are trying to conquer these genomes and also the epidemiology of the COVID-19.
Oh, okay. I think for the outbreak 17 years ago, okay, at that time, it is very difficult. Just because if you want to do sequencing, you need to have a pair of primer to amplify the DNA first, and then do a second sequencing, okay? But if we don't know the sequence, how can we design a primer? So that the problem 17 years ago, okay? So at that time, we use a shotgun sequencing, okay? So that's the reason why we take a long time. But mostly recently, with next generation sequencing, we can simply extract the DNA and then you do the sequencing, okay? Very simple. The most difficult will be the first step, okay? How to manipulate the virus safely, okay? Because we need to inactivate the virus, okay? So that it could be manipulated in the lab. Otherwise, the personnel will be infected, okay? This will be the first step, okay? The inactivation step, okay, will be the first bottleneck. And then the analysis, it will be another difficulty, okay? Just because there are more and more sequences, okay? And also there are more and more tools that could be used for analysis, okay? Then because of this, okay, we need to master a lot of skills. Unlike previously, okay, we can only use such simple method, okay, such tables to look at the virus. Now we can't, okay. We need to use very sophisticated statistical method and bioinformatic tools to analyze 1,000 or even 10,000 sequences at the same time, okay. And also a lot of information. Eventually, people are trying to use artificial intelligence to build models for the viral spread, okay? So the sequencing is simple now, but the analysis become very sophisticated.
Okay, thank you so much for your question. So here is a question from Dr. Vikas Sharma. So he is asking, does the genome of COVID-19 make a person immune or prone to the infection?
Sorry?
Does the genome of the COVID-19 helps the people to get immune or even prone to the infection? So I think what he's asking is about if you are being infected by COVID-19, will you have the immunity towards the COVID-19 itself? Since there's actually a lot of cases and there is a debate about whether after people got infected by COVID-19, and there's a debate whether there is going to be immunity over it. Yeah, thank you.
Okay. Well, very good interpretation of the question. Okay, so whether when you recover, okay, could the antibody protect you from another infection? I can say yes. Okay, so you could be reinfected by another virus that is similar, but that can escape from the antibody. But even if you could be infected, okay, your response will be lower. Just because although the antibody does not exactly match the virus, somehow it can partially suppress the virus. Okay, so unless the virus have mutated a lot, okay, that means that there are very substantial mutation. But as far as we know, SARS coronavirus or SARS-CoV have a pretty low mutation rate. Okay, so once you have been recovered, the chance that you will be seriously affected by the virus again in the next 2 years is pretty low.
Okay, thank you. So here is another question. Vika Chan raised her hand, so I'll unmute her to ask a question.
Hi, Professor. Thank you very much for your time. I'm actually a student with your genomics, Masters in Genomics program at CUHK, so it's very good to see you here. My question is, I've noticed that you said that there's a lot of advancements in sequencing technology over these years, the speed, the accuracy, and the analysis. But it seems that, so when you want to produce a vaccine, is there any way that bioinformatics could actually be used to help speed up, I guess, the research and development behind these vaccines?
Okay. Actually, for the development of vaccines, there are different phases. There are phase 1, phase 2, phase 3. Because of the advancement of the sequencing and as well as bioinformatics, now we can use very good software to do the docking, okay, so that we know which epitope or which part of the genome is best for the development of vaccines. Okay, that can speed up the phase 1. Okay, so very recently there are already a lot of candidates of vaccine. Okay, because of the advancement in the sequencing, bioinformatics, and also as well as the structural analysis. But why the vaccine, okay, will emerge later, okay, just because clinical, so this is a problem, not the technology, but economics. You can imagine, okay, if they don't have an extensive phase 1 trial to test the safety of the vaccine, and then when they launch the vaccine and cause the death of patient, every patient, they may ask for the compensation of millions of US, okay? So if that's the case, no matter how many tube of vaccines they sell, they will lose, okay? So the reason why the vaccine will come even early next year will be the safety concern, okay? There should be, first of all, testing in a particular country, and then from a city first, okay? Then to a country, to a different population, okay? Unless they find that it works, in most population, and it is safe to most population. That takes a long time. That part cannot be shortened. Understand?
Great. Thank you very much.
You're welcome.
Okay, so here is a question from Dr. Vikas Sharma. So he said that you mentioned that there are a lot of various genome studies across the world, and he's wondering whether COVID-19 will be more infectious or more prone to any group or ethnicity.
The last part,
yeah. So
whether it is
whether it's going to be more infectious to a particular group, like a Chinese ethnicity or English ethnicity. Thank you.
So basically, the UK launched the biggest project. I think I don't know whether you know, there are human genome sequencing projects in different countries. Okay, some project in US, some in UK. Hong Kong will start a project soon. So in UK, recently, they are trying to correlate the genome, okay, the viral genome, with the human genome, see whether there are some special combination that will make the patient more severe, okay, have more severe symptoms, okay. So this is possible. I can say why different population may have different presentation of the disease, because for the human genome in different population we have SNPs, okay. Between individuals, we have around 3 to 4 million SNPs, okay? Some of the SNPs are functional, and those SNPs could be related, for example, to the lung function, to the immune function, okay? Or those will somehow affect how we deal with the viral infection. So if you ask me whether a virus will attack a particular population, I will say no. But if you ask me whether the virus have a more severe effect on a particular population, I will say yes. According to the history, okay, the most terrible effect of virus will be on native population, okay? Just like long time ago, the native US population, okay? Just because of the infected by the foreign virus, because just they live in a village for many years, many hundred years, okay? They have not, they do not have a chance to contact a lot of viruses, okay? Therefore, they are naive to most viruses and don't have the antibody. Once they are infected, okay, they will have a very severe response. Just like I can tell you some story during SARS, okay, that may be also very interesting. SARS virus caused a lot of patients in Hong Kong, am I right? But people suggested that this is originated from markets in Guangdong. But when people go back and look at those people work in the market, they have the antibody, but they have no symptoms. If you ask me why, just because during their life, they have a lot of chance to contact similar virus, okay? Just like if we have the chance to contact previous coronavirus outbreak, okay? Then although the virus changed, the response will be much smaller, okay? So if you ask me why, I can say in Guangdong, they will go to the wire market more frequently than Hong Kong, am I right? I seldom go to those market with live animal, am I right? They go there quite frequently, am I right? And eventually they may have some sort of immunity against many viruses, okay? So that's the reason why people also suggested that when the virus transfer to US, it may have more serious symptoms. Just because in US, they can never see live animal, live chicken, live pig, they can never see, okay? All the meats have been generated from factory, okay? So that may also make a difference in different population. May not because of the genetics, but because of the habits, Okay, so there are many factors that can affect the different response and effects to the virus.
Okay, thank you so much. So we will have one last question from Chan Hor Q, and I'll unmute you to ask questions.
Hello. So my question is that how are flu vaccines able to change every year? As in, how are epidemiologists able to predict what kind of flu strains are more prominent in each year? And to apply this into SARS and COVID, if we had developed vaccines against SARS back in 2003, would it have mitigated the COVID situation this year? And also, is there a risk of COVID, SARS-CoV-2 mutating so fast that even if we do develop a vaccine, after a couple of years, maybe it doesn't provide much protection anymore due to mutation?
Okay, thank you. So starting from the SARS-CoV up to now, okay, there are a lot of different outbreaks. I think you still remember the swine flu, the avian flu, a lot of outbreaks, okay? And in every outbreaks, people prepare vaccines. But at the end of the day, none of them have really helped, okay? Just because of the time for the development of the vaccine is too long, okay? The vaccine come, okay, later than the outbreak, okay? So if you ask me whether the vaccine could help us for this outbreak, it depends on how long will this outbreak last. If this outbreak lasts for 2 to 3 years, this vaccine can help. The mutation is not a big problem. Mutation is not a big problem. Okay, just because even if just like for influenza virus, okay, every year we have a prediction. For example, H2N1, H3N5, something like this. Okay, even if you injected the wrong vaccine, that means not the exact type, that can still protect you, okay, and lead to a less severe consequence. Okay, so my simple answer will be, if the outbreak lasts long enough, okay, the vaccine can really help.
Okay, so thank you for answering. And since time is running short now, so this will be the end of today's webinar. So if you got any more questions, you feel free to email us at register@hklss.org. The same email you registered for this webinar. We will then compile them all together and help you forward to Professor Tsui for his advice. Once again, we sincerely appreciate Professor Tsui for sharing insights into the COVID-19 today. And thank you all for joining this webinar. Before you leave, remember to like our Hong Kong Life Sciences Society Facebook page and follow us on Instagram. Stay tuned with us, and our next webinar will be on the 10th of June, where we will have Professor Dennis Wirtz from Johns Hopkins University to share with us an overview of his research in cancer metastasis and the ongoing actions taken by Johns Hopkins in response to the COVID-19 pandemic. Again, thank you so much for joining. Thank you so much, Professor Tsui, and we look forward to seeing you again. Thank you so much, Professor Tsui, and also thank you so much for everyone for listening to this webinar.
Thank you.